Hello everybody. In this lecture, I'll talk about the development of the human brain. And as you know that early life experience may significantly alter the brain functions. And many studies have shown that early sensory experience um, can influence the development of the brain as well as can be related to many long-term disability of the brain functions. Therefore, it is very important that you know how the human brain is developed or formed from the very early stage of life. Okay, so first we will talk about the germ layers. So you know that after fertilization, the fertilized egg, which is called the zygote, that zygote, um, this is the fertilized egg, which is a single first cell of a new life, and then it divides by mitosis, right? So from one cell to two cell, then that way uh, the cell divides into many cells, and all these cell divisions are mitosis. Okay, and then we get a solid ball of like 16 to 32 cells. That stage is called the morula, and then what happens, this morula moves into the uterus, and when it enters into the uterus of the mother's body, what happens, this solid ball gets a cavity inside, and most of the cells, they get together to form a solid mass here. This is called the inner cell mass. Okay, and this outer shell is formed by the cells. These are called the trophoblasts. Cyto, also called cytotrophoblasts. And this inner cell mass becomes the embryo. So the inner cell mass is very important, because from the inner cell mass embryo is formed. Okay, so we'll see what happens in the inner cell mass. Inner cell mass forms three germ layers. So I'm drawing here again, and what happens, these cells, they line up to form three layers: ectoderm, endoderm, mesoderm. Okay, so this is one cellular layer. Okay, so this is the ectoderm, and then we get another layer, which is the middle layer, which is the mesoderm, and these are the germ layers. So this is the mesoderm, and the innermost one is the endoderm. Endo means inside, endoderm. So these three germ layers are formed. Okay, so ectoderm, mesoderm, endoderm. And from these three layers all body parts are formed. Okay, and the inner cell mass, these cells are pluripotent. Pluripotent cells, that means these cells are very early stage of stem cells, and they can be guided to move towards any direction to form any types of cells of the body. You know, human body has more than 200 different types of cells, and you can guide these cells to, you know, form or produce different types of cells, and that's why it is called pluripotent cell. And a lot of stem cell research actually are done by using these stem cells, very powerful. Okay, and this ectoderm forms the brain. Okay, so the brain structures are formed by this layer, ectoderm. Okay, so we'll see what happens in the ectoderm. So first you see here, this is the ectoderm shown here, and the cells of ectoderm form three parts in this layer. Surface ectoderm, this part, and then the crest, future neural crest, these cells will form the neural crest, and the middle portion, these cells form the neural plate. So surface ectoderm and neural crests, future neural crests and neural plate. And then some configuration change takes place. This middle portion invaginates, go inwards, and like this, the middle portion goes inwards and eventually gets separated and form the neural tube. So this is the neural tube formed by the neural plate. Okay, so this is the neural tube here, and you see that future neural crest, these cells here, they will form the neural crests, and then this part and this part, they will stay superficial outwards and form the surface ectoderm. Okay, and that neural tube, see here, the same thing, the surface ectoderm, neural crests, and the neural plate is invaginating to form the neural tube. And when the neural tube is formed, it looks like this. So you can see how a neural tube looks like, and eventually two openings are formed between day 24 to 26. Okay, so day prenatal, when the embryo is in very early stage, 24 to 26. Okay, so two neural pores are formed. This is the cranial pore, that means the opening, and this is the caudal pore, the bottom one. Okay, and then the neural tube, the configuration changes and it forms three pouches. Okay, and the top portion is called the prosencephalon. Okay, this part, and the middle portion or middle pouch is called the mesencephalon. Okay, and the bottom part is called the rhombencephalon. So prosencephalon, mesencephalon, and rhombencephalon. In simple terms, we say forebrain, midbrain, and hindbrain. Okay, and these three pouches, inside those pouches you have the cavities. Okay, and this is the most important stage in the early transformation of the developing brain. Okay, and these pouches are also called vesicles. You can say vesicles. Three vesicles are formed. Okay, and then from these three vesicles we get all the different parts of the future brain. Okay, so further you see more groups are formed, and if you see this one I just showed you before, three vesicles, and now if you see this right one, at around day 32 we see further changes. This is day 28 and this is 32 here, so you see some more changes take place. So the prosencephalon or forebrain part divides into two portions. One is called telencephalon, another is called diencephalon. Okay, this is telencephalon, this is diencephalon, and from the telencephalon we get the cerebral hemispheres. Okay, the largest part of the brain. And you know that hemisphere, there are two hemispheres, right? So they are separated by this groove here. Eventually they will get separated totally. And then from the diencephalon part we get the thalamus, hypothalamus, and as well as the pituitary. So thalamus, hypothalamus, pituitary, those structures are formed from this part, diencephalon part. And then remember, the thalamus is a very important structure in sensory processing. Okay, almost all sensory signals are processed in the thalamus. And then the middle portion becomes the midbrain. Okay, so this part, mesencephalon part, forms the midbrain, and then if you see the rhomboencephalon, the hindbrain part, it divides into two pouches or vesicles again, and lower one is myelencephalon. Okay, and the upper one forms the pons and cerebellum, and the lower one forms the medulla and the spinal cord. Okay, so that's how we get different brain parts from these three vesicles. Okay, and still you see this is a very early stage. Usually we say embryo when the period is 2 to 8 weeks of life. That means very early part, and before 2 weeks, first 14 days we say pre-embryonic period. Okay, so this is kind of early embryonic or almost middle embryonic period. We get all these structures. So a significant brain development is taking place during the prenatal. That means in the fetus, embryo, and then in the fetus. And when the baby borns, the brain gets a lot of structures already there. And any genetic disorder or any traumatic experience during the early brain development can cause, as I mentioned, the significant malfunction of the brain or disorder of the brain. Okay, so during this very early development of the brain, a number of factors can cause the deformities or disorders, developmental disorders. One is of course inheritance, mutant genes, chromosomal abnormalities can also lead to developmental disorders, and some syndromes. Those are usually related to the chromosomes defect, and other factors including lack of certain nutrients can also cause lack of brain formation or development, or defective brain development. Okay,